Hello, Dr. Robbins. You are a professor of biochemistry, molecular biology, and biophysics at the University of Minnesota, where you run a lab studying the biology of aging, including senescent stem cells and exosomes. So, welcome back to Modern Healthspan, and thank you so much for joining us today.
Great. Please be back, Richard.
So, Dr. Robbins, so we spoke about 4 months ago about exosomes, which was a great deep dive, really helpful. So, I have linked to that for anyone who is interested. But another area that you research is senescent cells and senolytics. And removing senescent cells has been kind of one of the areas of study for kind of anti-aging or longevity. And recently there was a paper from Dr. De Magalish, Senolytics Under Scrutiny in the Quest to Slow Aging. And he asked some questions about how successful senolytics were being at the moment. So it'd be great to have kind of an update of where you see the field of senescence cells and senolytics, because I think they are, a central part of kind of longevity. And maybe what we could start with is, so like a few years ago, we spoke to yourself, but we also spoke to like Dr. Kirkland and some others. So what has changed? But that was about 3 years ago. So what has changed about the thinking of senescent cells, do you think, in the last 3 years?
So 3 or 4 years ago, NIH put a lot of money towards an effort to identify, characterize, and spatially map senescent cells with aging. And I think many of us went into that effort thinking that senescent cells were fairly well defined. There weren't that many types of them. And if you cleared a senescent cell that expressed a marker like P16, that would be beneficial for many different diseases. But I think what we're learning is that senescence is very heterogeneous, that even with a specific cell type in the body, there are different types of senescent cells that arise by different types of stress. And that can actually express different types of inflammatory factors that can contribute to disease or aging in different ways. So I think it's become a more complex process. The way I envision this is it's going to turn out to be just like cancer. Cancer is very heterogeneous. Senescence can be very heterogeneous. And we really have to identify which cells we need to target to see the best effect on aging and age-related diseases. And I think the more we learn about senescence, the more we realize we don't know very much about it. And there's much more to understand in regard to identification and characterization of these cells, where they arise with age and what their contribution is to disease and aging.
Interesting. So how important is removing senescent cells for kind of rejuvenation or longevity? So I remember watching a talk by Dr. Vittorio Sebastiano about partial reprogramming. And one of the things he said is, yeah, we can partial reprogram all the cells, but you need to also remove the senescent cells because they're bad and they'll still be hanging around. So if we do everything else, do we still need to do the removing of the senescent cells as, to be able to get rejuvenation to work?
Right. So if you look at what's been published on partial rejuvenation from a variety of laboratories, Manuel Serrano, Juan Batiste Belmonte, that there's evidence that rejuvenation or partial rejuvenation can reduce the senescent cell burden in mice. So if you look at mice where you turn on Yamanaka factors, the OSKM factors, you actually see a reduction in senescence, whether these are the senescent cells being rejuvenated, or if you rejuvenate other cells that affect senescence, this still isn't clear. But it's my view that even if senescent cells are not the ones you want to target, the inflammation caused by senescent cells dampens the ability of stem cells to be functional. So if you're trying to rejuvenate stem cells in vivo, you clearly want to reduce the inflammation that is being driven by senescent cells. And some of the original papers showed that if you cleared senescent cells in the transgenic mouse model, the endogenous stem cells became more functional, more active, which will lead to better repair and potentially a longer health span. So I think the answer is most likely we would like to remove senescent cells, even with partial rejuvenation, but the partial rejuvenation itself may also reduce the burden of senescent cells. And so that has to be looked at very carefully. But the answer is, I think reducing the senescent cell burden would be beneficial across the board for other types of therapies to extend health span.
Right. Okay. So there were some trials that Dr. Magalesh mentioned in his paper.
Right.
And one of them was the ITP test with Fisetin, right?
Right.
So that was interesting because it didn't show extension of life. And in fact, it didn't even show a reduction in P16, which may be the same, may be related. So I just wondered, what were your thoughts on what happened there and why it didn't work?
So as a co-author on that study and a co-author who helped prepare the final manuscript, there's a lot of discussion about this data. So a negative result doesn't necessarily mean that the drug doesn't work. And there have been many drugs in the ITP, such as metformin, that did not show an extension of lifespan, despite the fact there's a lot of evidence in humans that metformin has benefits to extend health span. I think what we learned from that study is, one, that the mouse model that's used, these are the Het3 mice, which are very heterogeneous, supposed to mimic kind of the genetic background of the human population being very heterogeneous, that not all the mice in the group developed the same level of senescence. So senescence was very scattered between the mice. And so it was hard to show a significant reduction in P16. There was a trend towards a reduction in P16, but it wasn't significant. And part of that was only the 10 mice which we analyzed molecularly for effects on senescence. And so the speculation was, one, we didn't know if there was even enough Fisetin was being put in the food. It wasn't given at a high dose. It wasn't given kind of intermittently at a high dose as you would in normal senolytic and clinical trials. So the questions about the drug dose in the mice. But that aside, I think it means that with the mice you would like to treat are those with a high senescent cell burden. Just like in the human population, there's going to be a fraction, whether it's 10% or 50%, that's going to have higher senescent cell burden than others. And those are the ones that would respond to the senolytic. And I think that was the same in this very heterogeneous mouse model. There were some with high P16, some with low P16. And if you could break out all the data, I think there might have been more of a benefit in those mice that had high P16 to start. But that was not how the study was designed. So I think it's not necessarily a negative result. It's just not a positive result. And it means that we would have to either go back to the drawing board for the Het3 mice, have to design an experiment, or make more optimal senolytics, because Fisetin is not a very strong senolytic. It's a natural product. It appears to be relatively safe. It's just not very efficient. So we call it a very first generation senolytic, but can be improved on, we think, to improve the activity in mouse models such as this Het3 mouse model of aging.
Okay, so we think that Fisetin was removing at least some of the senescent cells, but it was the heterogeneity of the kind of the mouse model that really confused. Yeah, so that it became non-significant in terms of what was
right. But I think if we had analyzed many more mice, there would have been significance at reducing some of the senescent cell markers. It just, the way that it was designed, the way the ITP works isn't to do molecular analysis on hundreds of mice. It's to do lifespan extension on hundreds of mice and sacrifice a few for molecular analysis. And what we just observed was there was a lot of heterogeneity in the senescence markers we were using, which was only a few. But what we can measure, some mice had high senescent cell burden and some had low, which you might expect for a genetically diverse background of mice.
Okay.
And I think as we're going to talk about, I think that translates exactly to what we're seeing in the clinical trials in different diseases in humans.
Right. Okay. So Unity Biotech did a couple of senescent trials using two different drugs that they developed. And earlier this year, I think their second one did not show improvement over either placebo or the standard of care. Right. And they have not continued with that one. Do you have any insight into what happened at Unity? I mean, it's a company, so it's maybe harder.
Right. I only have speculation based on the biology. Obviously, the field has followed Unity very closely. So Unity started to try to treat osteoarthritis. They used a compound that disrupts interaction between P53 and MDM2 with the hypothesis that if you increase P53 activity, that would drive cells to apoptose that were highly damaged. In some cell types, that activation of P53 actually drives cells into senescence. So it was probably not the drug I would have chosen to take forward. The problem when injecting things in the joints, and I'm involved in other approaches to treat osteoarthritis, is that drugs injected intra-articularly are cleared from the joint space within minutes. So my guess is if you give this very infrequently, it's not going to be very effective at reducing whatever senescent cells there were in that joint at the given time, because it was a kind of a hit and run. You inject it, it's there for minutes, maybe an hour, and then it diffuses out of the joint space. So I think the issue there was, one, maybe the wrong drug, and two, this local delivery where it's not contained leads to very low drug concentrations that are there for only a short period of time. So I think it was a suboptimal clinical trial design. And then they switched to a second drug. It's targeting an anti-apoptotic factor called BCLXL. And this drug may have some toxicity if given systemically. So again, they chose a local condition. This is diabetic macular edema, to see if they could do a single injection of this inhibitor into the eye. And the initial data published in Nature Medicine, I thought, was actually remarkably positive. Patients had better eyesight, better night vision, even a year out. But then they did the pivotal trial comparing this to the standard of care, which is an anti-VEGF antibody. And they saw similar efficacy at one time point, but not at another. But unfortunately, the one where the efficacy wasn't as high was the one that they selected as their endpoint. So it didn't meet the criteria as established in the clinical trial design, and therefore they halted that whole program. So my guess is the issue here, again, a single injection of something into the back of the eye where they're seeing effects a year out. That's, I think, remarkable in itself. But it's only a single injection. So how many of the senescent cells were cleared? They don't know. Were there other senescent cells that the drug couldn't reach that are contributing to this pathology? Probably yes. We know that senescence in adipose tissue seems to have effects on brain function and potentially other aspects of the neurological system at a distance, probably due to these SAS factors. So even if you get rid of the cells locally, there may be effects of senescent cells in other parts of the body that contribute to disease. So I think there were a lot of positives that came out of the trial, but unfortunately it did not meet the primary endpoint. And that program has been shut down. But again, I view this as kind of a glass half full or 3/4 full, but there were positive results with a single injection into the back of the eye, which had not expected they would see very much. So in some ways, I think it's very positive. And you have better senolytics or more continued delivery of senolytics, I think you would see a much stronger improvement.
So the lesson is that it's difficult to do human trials, really.
Yes, humans are not, if we use a single genetic strain of humans, maybe, but yes, very difficult. And if I could just add to that, I think there were two other trials that were done that also hint at efficacy. So there was one from Dr. Sandeep Khosla at the Mayo Clinic targeting bone strength and formation in postmenopausal women. And this was with a combination of D plus Q, which again is the first generation senolytic that I helped develop with Jim Kirkland years ago. This now has been in multiple clinical trials. And what they saw was that there was actually improvement in bone strength and bone formation in the top third of women, at least the top third in the senescent cell burden. So if you measure the senescent cell burden in all the postmenopausal women, the top third at the highest level of senescence is measured in immune cells. Whether that's the right way to measure it, we don't know. But that top third actually showed the best improvement on bone strength and bone formation. And then there was a second trial done by Miranda Orr and kind of early stage Alzheimer's, so looking at mild cognitive impairment. And again, those that responded the best were those that had the highest level of senescent cell markers. So I think what we're learning is that, one, we probably need more optimal senolytics. I think systemic delivery is probably going to be the way to go to get rid of the whole senescent cell burden. And then we have to really identify the right population, subset of the population that has this highest senescent cell burden, because they're the ones who would respond. If you have no senescence, a senolytic is going to have no effect. So, and not everybody's going to have the same level of senescence as they age. And this is going to be true with disease and what caused the initiation of that disease, like osteoarthritis or Alzheimer's. And so if we can identify that top third or top half, those are the ones who will respond most effectively. So I think, again, it's this half full, half empty view of the clinical trials. I think there's been some very positive aspects, which I think warrants further development of better senolytics, and then identifying the right patient population. So, having said all that, the commentary that Joao wrote was, I mean, it was very insightful. I don't disagree with anything he says. I think he was trying to be provocative, because the data has not leapt out saying, yes, these are showing remarkable results in humans like they have in mice. But for the caveats I just went through, I think that it's still too early to make firm conclusions about the future of senolytics.
Yeah, we talked to other people as well. I mean, Dr. Andrea Meyer about the whole, giving a fixed dose to people, and you don't know how it affects those people. And so instead of titrating the dose based on how they react, it's maybe the wrong way. But anyway, that's another thing.
But I mean, the way they're talking about designing trials is actually each patient is his own control. You can start at a low dose and then work up in dosing and look at responses. And maybe that's what we'll have to be doing for many individuals, especially when it comes to kind of slowing aging. Really maybe a dose titration to see where the sweet spot is for a person that we know has an increase in senescent cells in their body, a measurable increase. We may have to do titrations because everybody's going to respond differently, just like the mice that were published where Fisetin did not have a statistically significant effect on lifespan.
You mentioned one of the trials that the Mayo Clinic was working on, which is this bone strength one. But I remember when we spoke with Dr. Kirkland, there were a number, some with Fisetin and some with this dasatinib and quercetin.
Right.
Did any of the others finish? Do we have any data from them?
So there's been 2 published studies, which you're probably well aware of. One for IPF, which is a devastating disease that once you're diagnosed, it's usually a 5-year death sentence or earlier. And so they saw minor effects, but nothing dramatic in the IPF trial. But that is a tough disease to have to treat once the fibrosis is developed in the lung. It may be very difficult to reverse that. And then there was one looking at diabetic patients to see if they can improve renal function. But they were able to take adipose biopsies and show that D plus Q was reducing the senescent cell burden in the adipose tissue in humans. So it appears that there was what we would call target engagement being the senescent cell, and the D plus Q was reducing the level of those senescent cells in adipose tissue. But there's really no clinical endpoint to measure in those studies. This was a short-term effort, and I don't know what the final results showed. There are numerous trials ongoing. They're being run through the Translational Geroscience Network that's being run by Jim Kirkland. It was based in Mayo. He's subsequently has moved, or I should say recently has moved to Cedars-Sinai, and they're continuing those trials. So a number of those trials are reaching their final patient enrollment or their final data analysis. I've not seen updates, but there are numerous trials with D plus Q, numerous trials with Fisetin, and other approaches, including not even drugs or small molecules, but mechanical stimulation and such to see if you can affect the senescent cell burden. So I think there's going to be a flurry of papers coming. I have not seen an update So can't comment at this point. But I think there's going to be a lot coming. So if we did this interview in a year, we'd have a lot more to talk about. Hopefully in a positive way, but maybe not. We'll just have to see.
We will have to see.
Yeah,
Yes.
so we did a trial for in COVID patients, and we were doing very well with enlisting patients that had gone to the hospital with COVID symptoms to see if Fisetin was going to reduce severity going forward. Unfortunately, the vaccine caused a lot of problems with those studies, and so patient enrollment went way down, and so hopefully that will be completed soon. But it really, in a positive way, led to a clinical trial that was difficult to complete. But there are other studies going on for other conditions. So again, I think there's going to be a lot of data coming. And so I think we will know for the first generation Senolytics, which are not very effective, if there's even evidence of a therapeutic effect.
Yes. Okay. And I assume that they're like double-blinded RCTs. And so until everything's finished, you have no real view. Yeah.
Right.
Do you think we are any better at identifying senescent cells? Do we have any better biomarkers for identifying them?
Great question. There are several reviews being written by the SetNet consortium trying to describe progress made on trying to identify and characterize senescent cells. I think the complexity of these is that there are biomarkers which may be tissue specific or cell type specific. Everything we had done up to more recently has been kind of bulk analysis of tissues. But now as we're trying to look at this, you can find cells that have P16 and evidence of DNA damage, or a cell with P21 and these 5 SAS factors. And I think we're still trying to determine if there's a universal marker of senescence, and a marker like SA beta gal, although found in many types of senescent cells, is not the only marker, and it's not only induced in the context of senescent cells. So we really don't have one or two perfect markers. And I think what we'll end up with are identification of markers that are prominent senescent cells in different tissues or different cell types. So there'll be a spectrum of markers. There's just not going to be one that's consistent across the whole board for senescence in every tissue and every cell type within that tissue. Senescent endothelial cell in the blood vessels may be far different than the hepatocytes, or far different than the microglia in the brain, in the pattern of what's expressed. So hopefully when the SetNet consortium is done with this analysis of identifying and spatially mapping senescent cells, we will have a much better idea of how to measure that senescent cell burden.
Kind of reminds me of our talk about exosomes and how they were so much more complicated than I thought.
Yes.
Yeah. Okay. Is there any more kind of data or thinking about whether removing all the senescent cells, well, as many of the senescent cells as we can, is a good idea? And there was a couple of thoughts that I had to kind of think about. So one is, my understanding is when we're young, the senescent cells don't hang around. We have them, but the immune system clears them up. But also, there are cases like the lens in the eye, I believe, where cells do not get replaced. And it is better to have a senescent cell than no cell. So anyway, just your thought on whether, like, if we could, removing like all of them would be a good idea or not.
Right. So it's a great question. I'm always asked about this in the context of neurological diseases. So working on Alzheimer's, working on Parkinson's, working on cognitive impairment. The question is if you kill off a senescent neuron, is that good or bad? I think our data is suggesting that the cells that drive these conditions are potentially inflammatory or senescent astrocytes and microglia, maybe even oligodendrocytes. It's not clear if the neuron is indeed the target of the Senolytics which we're working with. But I think the data suggesting, at least the mouse data, is that in almost every case, every disease case I'm aware of, reducing the senescent cell burden provides a benefit. And so I think it's better to get rid of something bad than it is to preserve it because it's filling space or providing still a function. I think if it's inflammatory and it's releasing proteases, reactive metabolites, et cetera, certain microRNAs, bad extracellular vesicles, that it's better to get rid of those cells than it is to maintain them. But we'll have to see as this plays out. I mean, obviously, clinical trials in humans will be a lot different than the preclinical studies being done in mice and in non-human primates. But so far, there's no evidence that getting rid of some of these bad cells leads to worse pathology. The confusion, I think, is that there's evidence that there's senescent cells that contribute to certain processes like wound healing. And what we need to separate is what is a good senescent cell from a bad senescent cell. And when you're old or diseased, is there even such thing as a good senescent cell? Because I think the evidence is coming out that the senescent cells that come up acutely and disappear that help with wound healing, the skin and other tissues, that these cells are beneficial when you're young, but when you're much older, the bad senescent cells dominate. So getting rid of them in older mice and potentially in older individuals would be beneficial. But again, we'll have to see how this plays out in the clinic.
So we talked about Senolytics and kind of Gen 1 Senolytics, which were like Fisetin and quercetin, which, as you say, they're like phytochemicals and they have many targets, right? But I also saw that there are maybe different types of senolytics now, like peptides and monoclonal antibodies. Right. And so what is coming for Gen 2 senolytics?
Yeah, it's a great question because, again, we really have to determine what senescent cells we need to be targeting. And I think the data is coming out that there's not one senolytic or senotherapeutic approach that's going to target all the different types of senescent cells. But things that are coming, starting with small molecules, my lab and others have spent a lot of time optimizing Fisetin. So done a lot of chemistry on Fisetin, medicinal chemistry, and they've come up with analogs of Fisetin that are probably tenfold more active than Fisetin and have some idea of some of the targets. We think we understand how we're optimizing the cell-like activities by optimizing the ability to target certain proteins or enzymes in the cell. We and others have identified compounds that induce senescent cells to die by ferroptosis. And this is because many senescent cells have an increase in iron and an increase in reactive oxygen species that leads to lipid peroxidation, but then in turn leads to senescence in these iron-heavy cells. And we've identified compounds that will clear this. And so is killing cells by ferroptosis, or will it be killing a different type of senescent cell through this mechanism? These are interesting questions. And there's a company called Rejuvenation Life that has a small molecule that seems to induce ferroptosis also through a different mechanism. And they're in clinical trials at this point. So I think there'll be data coming whether inducing senescent cell death through a different type of apoptosis, in this case ferroptosis, will it be beneficial for certain conditions. We think that improving immune function would be beneficial. So if you can clear the senescent cells in the immune system, we think that would lead to then clearance of the bad cells. So ways to improve immune function, or to use antibodies that directly target the senescent cell. But again, it's limited by the proteins on the senescent cell surface and how specific they are to senescent cells. Like, for example, UPAR, which has been shown by Scott Lowe's lab to be upregulated on senescent cells, it's expressed on macrophages and other cell subsets. So again, not one perfect marker that you can develop CAR T cells against or use an antibody. So I think all these approaches are interesting, but what's going to make them very specific for senescent cells and to clear the right type of senescent cells, we still have to, I think, spend a lot of time in the laboratory sorting through this. And I should just point out, I mean, I think in the long run, there's not going to be one senolytic that cures or treats everything. I do think it's going to be a cocktail. And the cocktail may be given at different time points. And the cocktail may vary depending upon the disease or condition that's trying to be treated, which again will depend on the type of senescent cells which are present.
If we could rejuvenate the immune system, I mean, and people are working on that, like Dr.
Yes.
Fahey and others, would that solve our problem? I mean, if we just make the immune system younger, would it go away and kill all the senescent cells? Do we think? I mean, we haven't done it, so we don't know. But do you think it would work?
It's an hypothesis that I think is being tested in the context of animal models and hopefully in the clinic at this point. I mean, that's what we don't know is if you treat somebody with Fisetin or D plus Q, how much of that senolytic activity of those compounds is leading to an improvement in immune function, which then leads to further reduction in senescent cell burden. So we don't really know when you treat a mouse or treat a human with a senolytic, you don't know where all the effects are. So it may be that how D plus Q is giving these partial effects is through improvement of immune function. All these things are testable, but we really haven't even started to scratch the surface. I think if you can improve immune function, which may be very difficult to do it well, it would have a tremendous benefit. Will it reduce all senescent cells in the brain? Not so sure. Maybe immune privileged organs, maybe the eye would not be the best target. It may be that certain joint compartments will not be affected. I guess an example we talk about a lot because we've shown benefits in the context of the intervertebral disc. So in your spine, as we age, it loses the cartilage, which is a shock absorber, which is the reason why we get shorter. And some people develop severe back pain, and we think that clearing senescent cells would be beneficial. But the blood supply going to the disc is very, very weak. So, how many senescent cells do we have to clear there? If you get the immune system to function, will it actually infiltrate into the disc and clear those cells? We don't know. All interesting questions, but clearly improving immune function would be beneficial across the board, whether it's clearing senescent cells or clearing pathogens or saving us from the next pandemic. A better immune system would be a very positive thing.
Yes. So just a thought. So the like second gen, the improved Fisetin.2 that you're working on, I assume that would be like from the FDA's point of view, a drug, and you would need to go through approval.
Right. So that's the downside. That's the reason why going with natural products or FDA approved drugs and repurposing them is a much easier path to the clinic. But again, I don't think any of the compounds that we've identified from FDA approved compounds or from natural products are as effective as they can be. So what we're trying to decide now is, do you make the investment in taking something that works 5 or 10-fold better, make that investment, or just try to use 5-fold more of Fisetin, for example? Would that be beneficial? But where we want to go forward with it, I think the low-hanging fruit is actually to see if it reduces senescence in the skin and actually can slow skin aging. And so that's something that we're working on, and that's actually where Beta Life with their ferroptosis-inducing senolytic compound, their first study is actually for skin diseases. I think it's not necessarily to show that it's going to treat a skin disease like psoriasis or dermatitis, but they can measure if there's a reduction in senescence in the biopsies from those samples. So that's where they've started with their compound, because it was an easier process than to develop their compound for systemic delivery.
So you talked about there's many different types of senescent cells, and Senolytics may impact different kind of populations of these cells. So are you kind of working on that, on developing specific Senolytics for specific populations of cells? Or, are people thinking about that in detail at the moment?
Yes, I think people are thinking about that. I mean, we started off just using fibroblasts for generic screens of compounds, using senescent fibroblasts as the target. But we're realizing that you really have to do this for the specific tissue type or cell type within the tissue that we think is the key senescent or the key cell type that becomes senescent and drives pathology. So just to give you one example, we have a paper submitted where we actually tested the ability to induce senescence with different types of space radiation. So this was an effort to try to attract NASA to give us funding to develop drugs which might help keep Mars astronauts healthier during their long trip. So we've taken higher energy particles that are comprised of space radiation, silicon, hydrogen, iron, and compared them to gamma radiation or chemotherapeutic compound. And the senescence profile is different. So if you take a fibroblast and treat it with these different types of radiation, the gene expression patterns are different and the susceptibility of them are different. D plus Q works to kill some of those and not others. Conversely, Fisetin works on some and not others. Navitoclax, which is a BCL2 family inhibitor, works on some and not others. So depending even on the inducer of a single cell type to undergo senescence, changes the sensitivity of those cells to a senolytic, which really shows the complexity of this. If you're exposed to the galactic cosmic rays that are comprised of all these different high energy particles, you may need a cocktail to clear the different types of senescent cells that arise. So it's going to be very complicated. Now, if you had Jim Kirkland on, he would say, we don't really care about what cells are senescent. If you reduce the senescent cell burden systemically by 30%, 40%, regardless of what cells we're targeting, if you just reduce the inflammation caused by those senescent cells at X number, percent, that would be beneficial. So it's just, we don't care, just dampening the whole level of inflammation, the whole level of senescence is all we're trying to do. And that may be true. Maybe all are, dissecting down to a specific type of senescent cell and looking at susceptibility to a senolytic. Overall, we just want to induce the overall senescent cell burden to see a health benefit. And again, that's a question that can be addressed. Do we really need third generation senescent cell type specific senolytics? And I think the answer is probably going to be a combination. Reducing the overall burden is going to be good, but there's going to be certain cells which we really want to target to really give the best therapeutic effect.
Okay, so there's another thing we can do with senescent cells is we can use Senomorphics, right? So we can stop them producing some of these nasty chemicals. But what is the benefit of Senomorphics? I mean, it seems to me it's a lot harder because the senescent cell's still there and you have to kind of give it chronically. Right. So what are your thoughts on Senomorphics? Is anyone working on them?
Yes, I think there's been a lot of data published with Senomorphics. I think the best example is some of the work that we published, but others too, is that suppressing NF-kappa B, which is an inflammatory cytokine induced by stress, but it's upregulating senescent cells and helps drive the SAS. So using an NF-kappa B inhibitor, you can reduce the SASP, and we hypothesize it prevents the spread of senescence because you see a reduction in the overall level of senescence markers if you treat chronically with this Senomorphic NF-kappa B inhibitor. So here, you brought up the question about, is it bad to have specific cells killed that aren't replenished like a neuron? So a Senomorphic that suppresses the bad SAS factors would have the benefit of not eliminating that cell, but just suppressing the bad things that it makes. The downside is you have to treat with that Senomorphic much more frequently, which as a drug may make it much more appealing to big pharma because you have to give it, I don't know if daily, but have to give it frequently. So I think that there are reasons why certain conditions might be more suited for a Senomorphic. I would argue that aspirin itself probably reduces inflammation driven by senescent cells too, because it affects NF-kappa B. So there are things which probably do function like a Senomorphic. They probably do other things too. But one of the reasons why they provide some benefit is because they suppress inflammation and some of this inflammation from senescent cells that come up with disease and aging. So I think and rapamycin is another one that's been shown to extend lifespan in mice in a number of clinical trials, but it's been shown to suppress the SASP. And is it effect on healthspan and lifespan through the suppression of inflammation? Maybe, at least partly. So I think there's space for both. I mean, there may be, like I said, cases where you don't really want to kill off all the senescent cells, especially if you're killing off some good ones, which we don't know if we are with these drugs. But so, it could be you give a senolytic every so often and the senomorphic daily if they have different targets.
And it sounds from what you said, the way you're talking, that the Senomorphic may be more generic. It would impact a wider type, a wider kind of set of populations.
That's an hypothesis. I don't think it's been proven yet, but I think the pathways that lead to the upregulation of SASP, that has to be done by transcription factors. And there seems to be some which are more prominent than others. So STAT3, NF-kappa B, there's several others that targeting these should have a benefit. The question is there a downside? All these things have positive roles in certain types of cells. So, there's always a trade-off. But in theory, it may have a more general effect on the SASP than a specific senolytic.
Okay, one last thing I think is that may be possible is to reverse senescence. And is anyone trying to do, have we made any progress on that? Because before, I think the idea was that it's almost impossible.
Right. So, there's a new class of drugs. There's review articles talking about senoreverters. I don't know if they're different than Senomorphics, but people are talking about drugs that seem to reverse the senescent phenotype. My view that the ability to do this probably depends on how deep that senescence is. I think what we're learning is that when cells enter the senescent cell state, they go through different phases. So there's a cell that expresses different markers that may have been senescent for a year or 5 years. It's a deep senescent cell versus one that may have just arisen because you were exposed to radiation 4 days before. So reverting those back to a normal cell, I think may depend about how deep and how much damage there is to that cell. There's also discussion about, do you really want to make a damaged cell 100% normal? Because senescence arose as an anti-tumor mechanism when there's activation of oncogenes or extensive DNA damage. The cells that can't repair themselves undergo senescence and want to be cleared by the immune system. So do we really want to completely revert those cells? We'll have to see. These studies being done by Alto's labs and others trying to look at partial rejuvenation, that's something they're going to have to deal with, is what happens if you partially rejuvenate a damaged senescent cell or one that may have acquired an oncogenic mutation. In mice, we haven't seen any increase in cancer, but we'll have to see if there's any negative effects in human trials. But I think the concept is, if you're going to rejuvenate cells, you're probably going to end up rejuvenating some senescent cells too. So is that a benefit, or is that going to cause adverse effects? And so I think the jury, at least in my view, the jury's still out. People are surprised Senomorphics may not lead to a higher risk of cancer, but at least in mouse models, treating with these Senomorphics seems to extend healthspan and lifespan. So it doesn't increase cancer risk, it decreases it, or at least delays onset. But for some of these things that people are saying are reverting senescence, we'll have to see. I don't think there's been any long-term studies done in mice.
Yeah, so one thought I had on that was, my understanding is, like, when a cell begins to go wrong, it can go into senescence, or it could go into cancer, or it could die. They pop.
Right.
Yeah. But once a cell has decided what it's going to be, like cancer or senescence, then the senescent cell itself does not help with anti-cancer. It's just that the choice makes an option rather than going into cancer. So is that correct? So then just removing senescent cells should not impact cancer. Well, unless, like you say, like reverting it back to an earlier stage where it can make the wrong choice.
But so a couple comments, and there are people working on this, not myself, but there are others working on this, and the preliminary data is again showing it's more complicated than we thought. But the question is it the senescent cell that eventually leads to the cancer cell? Because if you have a senescent cell, it could be that it's acquired changes that allow it to break through senescence. So, is it that 0.001% of senescent cells that have all these mutations that somehow can break through that, mutated P53 or mutated whatever. So is it the senescent cell that actually leading to the cancer? Alternatively, is it the senescent cell in the microenvironment, the inflammation that may be driving other cells into a cancerous state? So people are using reporter mice where you can have when P16 and P21, which are markers of senescence, increase dramatically, turn on a marker gene, and then determine if the tumor cell actually has activation of the marker gene because that one time they had high P16 and high P21. Or is it another cell that arises dependent upon senescent cells? So if you clear senescent cells in the transgenic model with a senolytic, do you see the same rate of cancer coming up? And if the cancer cell doesn't have this fluorescent label, it means that it's not coming from a senescent cell, but does the senescent cell contribute? And these experiments they're being done in aging mice, they're being done in obese mice because there's a lot of thought that adiposity can lead to all this inflammation that drives cancer. And I think we're going to know, and I think it's probably going to be different for different types of tumors. Unfortunately, it may be some are derived from senescent cells, and some are driven by senescent cells, and some are not affected at all by senescent cells. So, very interesting questions that I thought would have been addressed very early on. I do think all the mouse data says clearing senescent cells reduces your risk of cancer or delays onset. I think the mouse data is very supportive of that. And my guess is in humans that reducing senescence is going to have a benefit for most types of cancers.
So one of the benefits of senescent cells is wound healing.
Right.
Is it a specific type? So like, is it senescent epithelial cells that do the wound healing? So if you knew what the type was, maybe you could kill the other types and leave the ones that do the wound healing. Would that be a strategy that you could pursue?
So, yes, I think that would be a strategy. There is a hypothesis that the beneficial cells that may not persist for as long don't upregulate the same pathways, anti-apoptotic pathways, as a chronic senescent cell. So there has been speculation that many of the drugs we have targeting these specific anti-apoptotic pathways may not affect the positive senescent cell, the beneficial senescent cell. What we don't know is what differentiates a beneficial senescent cell from one that has adverse effects, the chronic ones that come up with aging. We're still trying to sort through because we have no assays in cell culture to characterize these beneficial activities of a senescent cell. People are working on that, but I'm not aware of any clear evidence that you can delineate what's a good cell, good senescence versus bad senescence at the molecular level in cell culture. So some people speculated that these beneficial cells are not affected by many of the senolytics we've developed. But that still has to be tested. And then I think, as we know more about what separates good from bad senescent cells, we really can develop drugs that target one and not the other. But we're still have a very poor understanding of what a good senescent cell looks like and how it's different from a bad senescent cell.
In your lab, are you just looking at how you're reducing senescent cells, or are you looking at specific indications in mice, I assume, like to see whether you're improving that indication?
So what we have done historically in my lab is I work very closely with the director of our institute, Laura Niederhofer, who's made progeria models where these mice have more DNA damage, and so they have accelerated senescence and accelerated disease. And these mice develop every pathology that aging, natural aging mice develop, and most of the pathologies that humans develop. So if we treat these mice, and we can do it short term for 4 to 6 weeks, we can really look in what tissue or what condition is improved the most. And for some of the compounds, you see effects on the musculoskeletal system. On others, it's more neurological system. So we're kind of using this accelerated aging, which we think is driven predominantly by accelerated senescence, as a model system to see where these drugs, where they go, what they affect, and what type of senescent cells are cleared, and what pathologies are improved, at least age-related pathologies. We're not doing many disease-specific models. We're really focused on aging. But if you see improvement in the liver, or improvement in the spleen, or improvement in the brain of this accelerated aging model, it would suggest these drugs could work for conditions in humans of those organs. At least that's the hypothesis.
What do you see as the most, what's the most exciting things that are happening in the Senolytics or senescent cell fields at the moment? I mean, what's the kind of future for this area?
Yeah, I think, unfortunately, many fields, everybody's working on senescence now. It's kind of the hallmarks of aging. It's one that may be more tractable than others to intervene in. So everybody's screening for senolytics. So, the journals are full of new compounds that work in cell culture, maybe in an animal model. My view is like the gene therapy field where I used to work. You really have to move these things into the clinic and show how they're working, if they're working in the clinical setting, to really facilitate future discovery. I mean, we can screen for hundreds of more senolytics, we can optimize them, but we really have to go bench to bedside and back to the bench. So I'm really excited about seeing what comes out of these initial trials, and then how we should proceed based on the clinical data. So we're developing drugs that target endothelial cells, epithelial cells, skin fibroblasts, microglia, astrocytes, chondrocytes, and maybe many of these, some of these will be very effective in humans. But I really like to see how these clinical trials, because many of these conditions we're trying to treat or could treat, they're being tested clinically at this point. So I really think the translational aspect of this, of an ongoing, and with results coming in the next 2 or 3 years, are really going to have a huge influence on the field. But where I think we're going to end up is a cocktail. It's going to be a cocktail of targeting the key senescent cell types in the key tissue, and it's going to be not done with a single compound. It's going to require, to optimize this at least, it's going to require a cocktail. You may want to improve immune function, you may want to improve liver function, you may want to improve your brain function, and there may be different drugs that target those senescent cells in those different tissues.
Would you work with a commercial organization to take something to the clinic? I mean, human trials are expensive and tricky.
Which is a problem for the field, because Unity's failure, and again, I say failure because they didn't meet the primary endpoint and investors lost confidence, but I actually thought there was the positive results coming out of their trial. But Unity's failure has caused big pharma to back off. I mean, that's what all it takes is 1 or 2 non-positive clinical studies in big pharma. So the answer is, for this to go forward, we're going to need big pharma. We're going to need their investment. We're going to need the medicinal chemists. We're going to need the people who do pharm tox. We're going to need the regulatory people. And so hopefully the clinical trials that are ongoing are going to restore interest and provide some confidence that big pharma should make this investment. So, I mean, that's the problem with designing trials that don't have the great chance of working, I think, like injecting something into the joint, is that it can lead to a negative, has a negative impact on the pharma industry. And that slows progress due to the financial aspect. And we'll have to see what happens with NIH going forward, if funding levels are cut. that will also have an effect on the clinical trials that are being done by NIH-sponsored investigators.
That is true. We'll see. But I mean, it will be very interesting to see what happens with these kind of, I guess, smallish trials that are ongoing, and see what those results are.
So Jim Kirkland keeps alluding at meetings, and I'm going to see him in 2 weeks at the ARDD meeting, of all the positive results that are starting to come out, but he's not the PI on them. He's just kind of coordinating them through his lab, helps with a lot of the assays. So he can't talk about them publicly. They're not his studies, but he's talking about positive results that are soon to be released to the public. So I'm hopeful in the next 6 months, we'll see a lot of data coming, which hopefully some of it will show that Senolytics are engaging their targets and are providing improvement in pathology. My fingers are crossed.
Yes. Yeah. So where can people follow your work? And I assume that if they're looking for these other trials, and the only thing is to kind of scan PubMed or set up
Semantic
I
think
Scholar.
the best place to look for those is actually if you search for the translational Geroscience Network, so TGN, Translational Geroscience Network. If you Google that, there's a website, and for people who are not members of it, there's still information about ongoing clinical trials. There's not data, but there's ongoing clinical trials that TGN, of which I think there's 80-something trials. They're not all Senolytics. There's many other types of compounds which are being tested. But that information is there. I am not the best at keeping my website up to date. We've talked for a while about sending out a newsletter, providing a newsletter quarterly, kind of summarizing what our institute is doing, including what my lab is up to. We haven't done that, but I think this year we're finally going to launch that. So that would be something to look out for, that you'd be able to go to our website for Institute and download the newsletter. We are starting clinical trials with Fisetin and several other compounds in the next year or so. We're also doing a lot of biomarker discovery. So like many others, to try to see if we can identify the right biomarkers that predict senescent cell burden. So I think keep an eye on our institute's website. There's going to be information coming. We are the Masonic Institute on the Biology of Aging and Metabolism. We have if you search for MyBAM, our website comes up and there's information there. We are now part of the Nathan Schock network of centers of excellence. So there'll be more information provided on that website too about ongoing studies. But I don't think that as an academic site, we or others have been as good as we should be about providing updates to the lay public, and we should be much better. And we're making efforts to correct that defect in our program and trying to put this newsletter together and provide more information to those who are interested.
Sounds wonderful. So thank you very much. And yeah, thank you so much for joining us again today. It was really educational, as always, to talk to you.
That's my pleasure, Richard. I always enjoy the questions you ask. It's an exciting time in this field, and I'm very optimistic about the future. But I hope to have a chance to come back in a year and talk about all the positive developments in the field.
Well, absolutely. Let me know when you have, or if I see, I'll reach out to you.
Thanks, Richard. Thank you very much.
Thank you.
Take care.
You too. Yeah. Bye.